So today we'll be discussing about two topics that are cerebral cortex and cerebellar cortex. Okay, that means cerebrum and cerebellum. Okay. Now these two topics are very important. Okay, and I will show you the slide also today. Uh, what I am planning is this slide of cerebellum will be kept for this practical exam. Identification of those slides are very easy. Okay, very easy. If you see once, you will identify. Okay, so that's why what I am planning is to keep those slides. Now, in cerebrum, we'll be studying about the cerebral cortex only. Now, the question is, do you know where the cerebral cortex is situated in the cerebrum? Mm-hmm. No. See, you know that two cerebral hemispheres are there, right and left cerebral hemisphere. Okay? Now, in one cerebral hemisphere, if you take any one, right or left, in that, where is the cerebral cortex situated? For your reference, you can see. For your reference, you can see the diagram. Okay. Now, can anyone tell me which section is this? Which section of brain is this? Which section of brain is this? What are the different sections of our body? Sagittal section, then transverse section and coronal section. So, which section is this? This is coronal section of brain. Okay, this is coronal section of brain. Okay, now in the coronal section you can see two color areas are shown. Can you see this dark color area and this bright color area? Why I have shown you this? So that you can understand that this is the cerebral cortex. This part. This is the cerebral cortex. Okay? So we have to study this particular area. The layers, microscopic structure, the layers of this area. So in case of cerebrum, the outer part is made up of cerebral cortex. What is present in the cerebral cortex? Gray matter is present in the cerebral cortex. And central part is made up of white matter, where the fibers are present. Deep to the white matter, the masses of gray matter, you can see some masses of gray matter are also placed. Okay, the same thing you can see in case of cerebellum also, but the arrangement is different. So, in case of cerebrum, you can see it is having a outer mass of gray matter and inner core of white matter. Okay. Now, gray matter of the cerebrum is called as cerebral cortex. Okay. Now, gray matter consists of cell bodies of neurons. जो gray matter होता है, it is made up of what? It is made up of cell bodies of neurons. Okay. Then neuroglia, processes of the neurons. Okay, and blood vessels. This is forming this particular cerebral cortex. The cell bodies of the neurons are there, processes of the neurons, neuroglia, cells and blood vessels. These are forming the cerebral cortex. And externally it is covered by a layer of meninges that is called as pia matter. Okay. Next, gray matter has numerous synapses between the neuronal process. So, though different types of neurons are present along with their processes, so they are also having synapses. Okay. Next, total 6 layers are there in the cerebral cortex. That means this particular area, what you can see here, microscopically, how many layers are there? 6 layers are present. Okay. Now, you have to understand one thing: the cerebral cortex is not homogeneous in nature. Okay. इसका मतलब क्या है? Cerebral cortex जो होता है, it is not homogeneous, it is heterogeneous. What is the meaning of this? Means the structure is not same. ठीक है, structure is not same. So there are some variations in the cerebral cortex. So cerebral cortex shows considerable variation. Okay, based on the structural variation, it is of two types. It is homotypical typical cortex and heterotypical cortex. Homotypical means those 6 layers are same, all the types of cells are same, okay. But heterotypical means the 6 layers are not well maintained or not well defined, okay. In heterotypical also further it is divided into two types, that is the granular cortex and agranular cortex. ग्रेन्युलर कॉर्टेक्स में क्या होता है, जो ग्रेन्युलर सेल्स है, नंबर ऑफ ग्रेन्युलर सेल्स आर मोर और यू कैन से दे आर वेल डेवलप्ड, दैट्स व्हाई इट इज कॉल्ड एज ग्रेन्युलर कॉर्टेक्स. कहाँ पे रहता है ग्रेन्युलर कॉर्टेक्स? दिस आर प्राइमरी सेंसरी एरिया, एक्वास्टिक एरिया, विजुअल एरियाज एक्सेट्रा, ओके. In these particular areas of the brain, the granular cortex is well developed, okay. Next, agranular cortex is there. Here granular cells are poorly developed, instead of that pyramidal cells are well developed, okay. That also larger pyramidal cells of pets. For example, you can see frontal lobe, primary motor cortex. These are the areas where the agranular cortex is well developed. So this is the variation. Same cerebral cortex, but variations are there. That's why the structure is heterogeneous, not homogeneous, okay. Next, based on phylogenetic scale, okay, based on phylogenetics scale, again it is divided into 3 types, okay. What are they? Archicortex, paleocortex and neocortex. Now what is the meaning of phylogenetic scale? Yes, on based on evolution, ठीक है. फाइलोजेनेटिक स्केल का मतलब है बेस्ड ऑन इवोल्यूशन. सो बेस्ड ऑन इवोल्यूशन, ओके, the cerebral cortex is again divided into 3 types, 3 categories: archicortex, paleocortex and neocortex. Archicortex is the oldest and constitute the sizable portion of cerebrum, and it is well developed in case of lower vertebrates. In human being, the archicortex is represented by hippocampus and rhinocephalic region. Okay, hippocampus and rhinocephalic region. These 2 parts of the human brain is representing the archicortex. Okay, rhinocephalic region is. Can anyone tell me what is the rhinocephalic region? It is the olfactory bulb, olfactory stria. All those areas are under rhinocephalic region. Okay. Now structurally, if you see, these are made up of only 3 layers. Next, paleocortex, it is the intermediate in development, and in case of human being, it is represented by cingulate gyrus. I hope you have studied about the sulcus and gyrus of brain. Yes. So the cingulate gyrus is only representing the paleocortex. Next is the neocortex. Phylogenetically, it is the most recent in development. 90% of our brain, okay, is made up of neocortex, okay, where all the 6 layers are present, okay. So in phylogenetic scale also you can see the entire cerebral cortex is not same, it is having some variations, okay. So which one we have to study? Neocortex. We have to study obviously about the neocortex. So how many layers are there in the neocortex? 6 layers are there. So for that, let me show you one diagram. So here you can see the diagram, okay. So which is the outermost layer? See, in case of, yes, listen, in case of cerebrum and cerebellum, the basic concept is only layers, nothing else, ठीक है. सेरिब्रम में 6 लेयर्स रहता है, 6 लेयर्स आर देयर, एंड इन केस ऑफ सेरिबेलम 3 लेयर्स आर देयर, हाफ. Like this you can remember: cerebellum 3, cerebrum 6 layers. Another way is there: cerebrum is larger, okay, that's why it got 6 layers. Cerebellum is smaller, that's why it got 3 layers. This is the way to remember the number of layers histologically, okay. So first of all, you know that in histology, if layers are there, it must be from outside to inside or inside to outside. Now here the layers are present from outside to inside, okay. So this is the outermost layer and this is the innermost layer. Now the question is, जो outermost layer है, outer to that what is present? जो outermost layer है, outer to that what is present? And जो innermost layer है, inner to that what is present? Yes, very good. हाँ बोलो. Yes, you are correct. Yes. No. हाँ, biometal. Very good. Okay. So see, like this you have to understand the things. देखो, these are the layers of cerebral cortex. In the beginning I have shown you the cerebral cortex. कहाँ पे cerebral cortex रहता है, right? So these are the layers of cerebral cortex. This is the outermost layer. So outer to this what will be present? The pyramidal will be there. Okay, the meninges which is covering the brain. Next, deep to this what will be present? White matter. Okay, fibers are present. Next, so we'll start from first layer, that is the molecular layer. Okay, so what is present in the molecular? In molecular layer, it is situated just below the pyramidal. Okay, next, predominantly if you see the molecular layer, it is made up of fibers. Along with that, some blood capillaries are also present. Now the fibers, from where these fibers are coming? These fibers are coming from pyramidal cells, stellate cells, Martinotti cells, horizontal cells, etc. Okay, so these are the cells from which the fibers are going into this particular layer, the molecular layer. Now, in molecular layer, only one type of cells are present. Okay, they are also horizontally placed, and that's why they are called as horizontal cells of Castle or Castle-Ridgius cell. CAJAL, Castle-Ridgius cells. Okay, only one type of cells are present in the molecular layer. Okay, so here I am talking about this one. So if you see this particular layer, it will appear brighter under microscope. When you will do the H&E staining, after that when you see the slide, this particular layer will appear brighter. Why? Because cells are not there, only one type of cells are present. Okay, mostly it is made up of fibers. Because of that, the appearance is brighter. Okay, because this is very important. Why this is important? When you will see the slide, okay, at that time these demarcations are not there. जो slide है उसमें देखो, for example, see this is one layer, this is one layer. In between both the layer, can you see a line is drawn? In histoslide, can you see such kind of lines? No. So clear cut demarcations नहीं है. Here the lines are given. This is a schematic diagram to make you understand that 6 layers are there. Okay, that's why you can see the clear cut demarcation. But same thing when you will see under slide, under microscope, ये lines दिखाई नहीं देगा. At that time how will understand कि ये कौन सा layer है, ये कौन सा layer है? How will understand? Yeah, based on the staining properties. Simple. At that time, based on the staining properties, you have to understand which layer is which one. So you have to know the staining property. So the first layer है, as I said, cells are not there. Only one type of cells are present. That also horizontal cells of Castle or Castle-Ridgius cell. Remaining, it is entirely made up of fibers. That's why it will appear brighter. As simple as that. Next layer, if you see external granular layer. Now here it is made up of densely packed stellate cell. So if a layer where densely packed stellate cells are there, what will be the appearance? Dark appearance. Okay, so here the appearance will be darker, and you can see many nuclei present in these cells. Okay, so this is the external granular layer. Now in this external granular layer, along with the stellate cells, you can also see small pyramidal cells. Okay, small pyramidal cells are also present. I am talking about this one, external granular layer. Here you can see densely packed granule cells are present. Next is the external pyramidal. Next is the external pyramidal layer. Okay, in external pyramidal layer, small or medium sized pyramidal cells are present. Okay, in external pyramidal layer, small or you can see medium size pyramidal cells are there. Okay. Now in this cerebrum, you can see different types of pyramidal cells are present. Okay. Small pyramidal cells are there, medium size and large pyramidal cells. Three different types of pyramidal cells are present. Okay. We'll discuss what is the importance of those pyramidal cells. Next is the internal granular layer. This is the internal granular layer. Here also closely packed stellate cells are there. But along with that, you can also see horizontal lines. Can you see the horizontal lines? Okay. So in the middle of the layer, horizontally arranged white fibers are present, and those are called as external band of Baylor. Okay, those horizontally arranged fibers, they are called as external band of Baylor, which are seen in internal granular layer. Okay, now they are also called as white stria. Now when you will see the visual cortex, okay, especially the occipital lobe, this white stria is very thick and very prominent in this region. Okay, that's why it is also called as striate cortex. Next is the internal pyramidal layer. Okay, internal pyramidal layer are also called as ganglionic layer. Here you can see large pyramidal cells are present, and they are also called as pyramidal cells of Betz, VETZ. Okay. Now these are the largest pyramidal cells, and here the fibers which are coming out from those cells, they are forming the internal band of Baylor. You can see these are the internal band of Baylor. Next to that, the deepest layer is the multiform layer. Okay, in multiform layer, different types of cells are present. Okay, for example, fusiform cells are there, Martinotti cells are there, stellate cells are there. Okay, like this, different forms of cells are present in the multiform layer. Okay, so we have encountered different types of cells. Okay, so you must know some important function of those cells. So first we will see about the horizontal cells. Okay. So horizontal cells are also called as causal reticulous cells, most superficial. Where are they situated? They are situated in the most superficial part of the cortex. Okay. Now they are very rare, and especially in case of adult brain, the number of these horizontal cells are very less. And if you see the function, the main function is the organization of developing brain. It helps in the organization of developing brain. Okay. And as I said, they are present in superficial layer only. Next, cells of Martinotti and fusiform cells. Now these two cells are present in the deepest layer. Now cells of Martinotti, these are inhibitory cells. Okay. Now inhibitory to what? They are inhibitory to pyramidal cells. Next, fusiform cells, they are spindle-shaped cells, and they are having some special features, or you can say special function. So they play a crucial role in the high order cognitive function, including attention, perception, memory, etc. Okay, that is the function of the fusiform cells. Next is the pyramidal cells. So 75% of the cell population in cerebral cortex is made up of pyramidal cells. Okay. Now these cells are very important, and depending on the size, okay, three different types of pyramidal cells are present. Okay, it is small pyramidal cell, medium pyramidal cells, and large pyramidal cell. The large pyramidal cells are also called as pyramidal cells of Betz or Betz cells. Okay. Here you can see the size, 10 micrometer, then 50 micrometer, 100 to 125 micrometer. So like this, different size of pyramidal cells are present. Now if you see the pyramidal cell, the axons of the pyramidal cells, mainly they are forming the projection fibers, which enters the white matter. Can you tell me what are those projection fibers? Only descending tracts. No. Projection in brain, as you know, that three different types of fibers are there. Among them, the projection fibers, they connect the brain, or you can say they connect the cerebral cortex to the subcortical centers. Yeah, that's why I said subcortical center, not only spinal cord. Subcortical centers. It includes brainstem. It includes spinal cord. It includes cerebellum. These are the subcortical centers. Okay. So that is the projection fiber. Have you studied about the internal capsule? Not yet. So when you will study about the internal capsule, at that time you will understand in detail about the projection fibers. Okay. Next is the stellate cell or granule cell. They are much smaller pyramidal as compared to the pyramidal cell. Okay. If you see the size, it is only 8 micrometer. Now what is the shape? It is star shaped. Stellate cell means star shaped. Now the appearance, if you see the appearance under microscope, it is just like a dust of cloud particles. That's why the cerebral cortex, where these particular cells are present, this is called as neocortex. ठीक है? मतलब क्या है? The cells are very small. ठीक है? If you see the shape, it is star shaped. The appearance under microscope is cloud dust of cloud particles. Because of the presence, the appearance is called as neocortex. The meaning of conios is also called as cloud. Okay. Now, what is the main function? The main function is they release GABA neurotransmitter. Okay. What is GABA? Gamma aminobutyric acid. Okay, that is a neurotransmitter which is inhibitory neurotransmitter in central nervous system, and that is secreted by these stellate cells or granule cells. Okay. Next, here you can see a diagram. In this diagram, all the types of cells, whatever we have discussed, okay, the diagram is shown over here. So the previous diagram you have to draw. Now next one is the schematic diagram and histological diagram, both under low magnification. So this is the schematic diagram and this is the histological diagram of cerebrum under low magnification. Now here you can see lines are not there. Okay. So in the first image I have shown you that lines can be seen so that you can differentiate, do the differentiation, but here you can't do the differentiation based on the lines. Okay. So how you will know that which layer is which? कौन सा layer है? So yeah, so this is the outermost layer. First of all, you can see pia mater is present. Okay. Next to the pia mater is the molecular layer. So what I have mentioned, molecular layer के appearance कैसा होता है? It appears brighter. Why? Because only one type of cell is present. Remaining, it is made up of fibers. Okay. So that's why you can see it is bright in appearance. Next, you can see external granular layer. Okay. See, so many granule cells can be seen. Okay. After that, you can see the external pyramidal layer. Now, in case of a pyramidal layer, small pyramidal cells can be seen very easily. Okay. Next, this is the internal granular layer. Here, this is the internal pyramidal layer where you can see the large pyramidal cells. Okay. So, as I said, three different types of pyramidal cells are there: small, medium, and large. So, here you can see the small pyramidal cells. Here you can see the large pyramidal cells. Okay. So, by looking at the diagram under low magnification, easily you can do the differentiation between the pyramidal cells and the granule cells. जो granule cells होता है under low magnification it will appear just like a dot because they are extremely small. You need high magnification to see the shape of the cells. Okay. तो ये जो छोटा छोटा dot दिखाई दे रहा है, these are nothing but the granule cells only. Okay. But pyramidal cells, these are larger cells. Easily it can be seen. ये देखो. So this is the large pyramidal cells which are present in the internal pyramidal layer. Okay. And lastly, this is the multiform layer. In multiform layer, different types of cells are present. Okay. Now here, I have not added the EM structure. As you know, each and every cell is having EM structure. Okay. That one I have not added. I have seen it is very difficult. और जो हिस्टो बुक है, द बुक विच यू आर फॉलोइंग, ठीक है, उसमें नहीं दिया है. इन अदर बुक्स इट इज गिवन, बट द बुक विच यू आर फॉलोइंग that is not given, so that's why I have not added the EM structure. Okay, that will make it very complicated. Okay, so what do you think, is it easy or difficult? Can you identify this in the exam? ये तो आइडेंटिफाई करना है, इसमें कुछ है ही नहीं. जो लार्ज पिरामिडियल सेल्स है, ओके, लार्जर पिरामिडियल सेल वो अच्छा से दिखाई देता है, ठीक है, तो वो नीचे देखना है कि पिरामिडियल सेल्स दिखाई दे रहा है कि नहीं. हाँ, तो आइडेंटिफिकेशन इट इज वेरी इजी. ओके, so that is all about the cerebrum. Next we'll discuss about the cerebellum. Now one thing which is easy in case of cerebellum is the cerebellar cortex is homogeneous in nature. ठीक है, that means no variations are present in the cortex. So in case of here also in cerebellum, cerebral cortex is there. The structure of cerebellum, first of all, it is uniform throughout the cortex. So it is also having an outer cortex and inner white matter. Outer cortex which is made up of grey matter. Inner part is made up of white matter. Now the substance of the white matter, in the substance of the white matter, masses of grey matter are present, and they are in the form of intracerebellar nuclei. 4 such nuclei are present. Okay, as you can see, dentate nucleus, then globus, emboliformis and fastigious. Okay, these are 4 different types of nuclei which are present. कहाँ पे रहता है? Deep into the white matter of the cerebellum. Okay, but they are masses of grey matter. Next, cerebral cortex, as you can see in the diagram, 3 layers are present. Outermost layer is called as molecular layer. That means outer to that you will be getting the pia matter. Middle one is called as Purkinje cell layer, and innermost layer is called as the granular layer. Deep to the inner layer, what is present? White matter is present. Okay? So if you see the diagram, see 3 layers are present. Here you will be getting the white matter. See, यहाँ पे white matter है. These are the 3 layers. From here white matter starts, and outside that you will be getting the pia matter. Okay. Now, before going to the microscopic structure, there is a important, you can say, gross aspect of the cerebellum. Okay. Now, if you remember the small intestine, when we have discussed about the small intestine, at that time I have told you that it is having villi, right? And we have studied the structure of villi also. Now, in order to study the structure of villi, we have taken one villus, right? Similarly, here the cerebellar cortex, okay, they are folded in such a way that, okay, so the cerebellar cortex is folded in such a way that cerebellum represents parallel transverse fissures. दो cerebral का जो cortex है, so in cerebellum also 2 cerebellar hemispheres are there. Okay, now those 2 cerebellar hemispheres, if you see the surface of the cerebellum, it is highly folded. It is folded in such a way that you can see the transverse parallel fissures. In between the fissures, a thin band-like structure is present. It is called as folia, F-O-L-I-A. ठीक है, so multiple folia are there. Here, suppose you have taken one folia. In that, you will see the center part is made up of white matter, and peripheral part is covered by grey matter. Okay. Here you can see the diagram. Can you see the transverse fissures? In case of cerebellum, it is folded in such a way that you can see parallel transverse fissures are there. So many parallel transverse fissures. Okay. Now, in between them, folia are present. If you take one structure of folia, that is the folium, then what you will get? First of all, you will be getting the central core, which is made up of white matter, and peripheral part is made up of grey matter. Here you can see this central core is made up of white matter, and peripheral part is made up of grey matter. Okay. Next, ये जो सेंट्रल व्हाइट मैटर है, it is not straight, it is in the form of branching pattern of tree. That's why it is called as arbor vitae cerebelli. Okay, that is called as arbor vitae cerebelli. Now why I am showing you this? Because in case of microscopic structure we have to take one folium, okay, and then we have to do the microscopic study, then we have to see the layers. So in microscopic structure you will see so many folia are there. From those folia you will be taking only one folium and then you have to do the study. So when you will take one folium, उस टाइम में क्या दिखाई देगा, जो सेंटर है उसमें वाइट थिन स्ट्रिप ऑफ वाइट मैटर दिखाई देगा, एंड अराउंड दैट यू विल बी गेटिंग द 3 layers ऑफ cerebellar cortex or 3 layers of gray matter. Okay. So the first layer is the molecular layer. Okay. The molecular layer always it will stain brighter. Why? Because here also will be getting number of fibers. Different types of fibers are there. Okay. And this layer is also situated just below the pia mater. Okay. Now here unmyelinated nerve fibers are there. Okay. From where the fibers are derived, as you can see, the fibers are derived from axons of cranial cells, stellate cell, basket cells. Okay. And dendrites of Purkinje and Golgi cells. So these are the different cells from which axons and dendrites are derived, which are making the molecular layer. Okay. So appearance is bright. In case of cerebrum, we have seen only one type of cell is there in the molecular layer. Here you are getting two types of cells. What are they? Superficial stellate cells and deep basket cells. Okay. These are the two types of cells present in the molecular layer. Superficial starts for S. Okay, stellate cell also start for S. So S. Okay, that's why the superficial cells are the stellate cell, deep cells are the basket cells. Okay. So as you can see, this is the first layer. So this is the superficial cells, stellate cell. These are the basket cells. And you can see all the different types of fibers which are present in this layer. That's why this layer appears brighter. Okay, eosinophilic appearance आएगा. Next, which is the next layer? Next layer is the Purkinje cell layer. Now, in case of Purkinje cell layer, only you can see one layer of Purkinje cells are present. It means not in multiple layers. Only one layer of Purkinje cells are present. And also the cell bodies of the Purkinje cells are present. Cell bodies. Okay. Now if you concentrate over here, so this is the Purkinje cell layer. Okay. Now here you can see the cell bodies of the Purkinje cell are present. ये देखो cell bodies of the Purkinje cell, cell bodies of the Purkinje cell. Okay. Only the bodies, cell bodies of the Purkinje cells are present. Single layer. Next, from the cell bodies, what is coming out? क्या निकल के आ रहा है? हाँ? No. What is coming out? You can see two things are coming out. One is going towards the molecular layer, another is going towards the white matter. ठीक है? So axons are coming out, along with that dendrites are also coming out. So here from the Purkinje cells, bodies of the Purkinje cells, the dendrites are going towards the molecular layer, as you can see in the diagram. Okay? Now once it is going towards the molecular layer, it is forming a dendritic tree. ये देखो, it is forming a dendritic tree. Okay, it is forming a dendritic tree. Next, what happens? इसका जो axon होता है, it is going deep. Once it is going deep, first of all, it will rely on the intracerebellar nuclei. So in the beginning, I have mentioned that जो white matter होता है, उसमें intracerebellar nuclei रहता है. So these axons will first rely on the intracerebellar nuclei, and then the fibers which are coming out, that will form the efferent fibers, sole efferent fibers of the cerebellum. Okay, cerebellum का भी efferent fibers है, efferent fibers है. Can you tell me what are the efferent fibers for cerebellum? Efferent fibers for cerebellum? It is climbing fibers and mossy fibers. Only two types of fibers are there. Okay, for if which are efferent to cerebellum. Those are climbing fiber and mossy fibers. ठीक है, तो efferent कौन सा होगा? That means which are coming out from the cerebellum. Those are the axons of these Purkinje cells. ठीक है, now those axons of the Purkinje cells, when they are coming out as efferent, they provides inhibitory feedback to this intracerebellar nucleus. Okay. Today, what you have studied in the first class? All those things are done. Okay. Now, so here you can see the dendrites of the Purkinje cell, as I said, where they are going into the molecular layer. Okay. Once they goes into the molecular layer, what they will do? They will synapse with the axons of the granule cells. So you can see they are synapsing with the axons of the granule cells. ये देखो, this is the granule cell का axons है. Okay, here they are synapsing. After that, they also synapse with the climbing fibers. So these are the climbing fibers. You can see they are synapsing with the climbing fibers. Okay. So once the dendrites are coming out, forming dendritic tree, it will synapse. So what it is synapsing with? So you can see it is synapsing with the climbing fibers and the granule cells, especially with the parallel fibers. You can see with the parallel fibers they are synapsing. Okay. Next is the granular layer. Okay, so this is the granular layer. Okay. Now granular layer में क्या रहता है? First of all, what will be the appearance of the granular layer? It will stain dark. Okay, because of the aggregation of granule cells. In case of cerebellum, along with the granule cells, another special type of cells are present. Those are called as Golgi cells, as you can see in the diagram. Okay, ये देखो. Now these Golgi cells, these are the largest cells of the cerebellar cortex. Short question. जो cerebellar cortex है, उसमें जितना भी type of cells रहता है, among them the largest one is the Golgi cell. Okay. Now this granule cell, what they do, the axons. Can you see the axons of the granule cells? These are the axons of the granule cells. So what they will do, they will pass to the molecular layer. ऊपर जाएगा. Once they reaches the molecular layer, they will bifurcate at T junction. And those, after bifurcation at T junctions, the fibers, they will run parallel to the long axis of the folium. And that's why they are called as parallel fibers. Okay. Parallel fibers. So this already we have discussed. Now in case of granular layer, in granular layer is stained dark. Okay. Now each granule cell give rise to 4 or 5 short dendrites. Okay. Appearance is claw-like, and the endings will synapse with the mossy fibers. And axon, you can see, reach the granule cell into the molecular layer, where bifurcates into T junction, where it forms the parallel fibers. Okay. Next, another type of cells are there which are large, but the number of cells are very less, and those are called as Golgi cells. Okay. Golgi cells are also having very important function. Now, if you see the location of the Golgi cell, the cell bodies are located just outside to the granular layer, below the Purkinje cells. Okay, below the Purkinje cells, there these Golgi cells are present. Hardly you will see any Golgi cell in the slide because the numbers are very less. Okay. Next, these Golgi cells, what they will do, they receive input from parallel fibers and mossy fibers, which excite the Golgi cell. Okay, parallel fibers and mossy fibers, from there they will receive the impulse, and which will excite the Golgi cells. Okay, then Golgi cells and recurrent collateral axons of the Purkinje cells will inhibit the Golgi cells. Now they give output dendrites to the granule cell, and what they will do, they will inhibit the neurons. Okay, Golgi cells are inhibitory neurons and diminish the excitability of the granule cells. The granule cells ko you can see that they are inhibiting. Okay, the excitability of the granule cells, they are inhibited by this particular Golgi cells. So no, no, no. No, 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 not Golgi cell, inhibit the granule cells. Okay, granule cells, because they provide inhibitory feedback to the granule cells, Golgi cells. Okay. Next, in white matter, so deep to the molecular layer, white matter is present. Three different types of fibers are there: intrinsic fibers, afferent fibers, and efferent fibers. Okay, intrinsic fibers means they are confined to cerebellum only. Now, in case of afferent fibers, two types of fibers are there: climbing fibers and mossy fibers. Okay, efferent fiber, already I told you, they are the axons of Purkinje cells. Now, in case of afferent, two types of fibers are there. Okay, climbing fibers and mossy fibers. So, what are these mossy fibers? All the fibers which are entering the cerebellum except the olivocerebellar fibers. They are called as mossy fibers. Now, these mossy fibers will go and synapse with the granule cells and the Golgi cells. Okay, that is the mossy fibers. Now, climbing fibers, they are derived from inferior olivary nuclear complex. Okay, inferior olivary nuclear complex. From there, the fibers which are coming, they are called as climbing fibers. Okay. Now, in molecular layer, now they pass to the granular layer and Purkinje cells and reach the molecular layers. Okay. So, in molecular layer, the collaterals of climbing fibers synapse with all the types of neurons which are present over there. Okay, that is the climbing fibers. So, two different types of afferent fibers are there: mossy fibers and climbing. Mossy fibers means all the fibers except olivocerebral fibers. Olivocerebral fibers. Okay, they are called as mossy fibers, which will go and synapse with granule cells and Golgi cells. Climbing fibers, the fibers which are coming from inferior olivary nuclear complex. Okay, what they will do, they will pass to the granular cells. Okay, or you can say granular layer. They will reach along with the climbing fibers in the molecular layer. Okay, along with the collaterals of the climbing fibers, and then synapse with all other types of cells which are present. That is the climbing fibers. Now, an important structure is formed that is called as glomeruli. As you can see with the mossy fibers. तो mossy fibers का जो terminal part होता है, that is dilated and that is called as rosette. Okay. Now that is synapsing with the axons of Golgi cells, and you can see axons of the granule cells, which is covered by a capsule, and this particular structure is called as glomeruli. Okay. Next, this is the histological diagram. Okay, this is low magnification, this is under high magnification. So what you can see, यहाँ पे क्या दिखाई दे रहा है? So what does it called as? This entire part, folia. So this is one folium. This is one folium. This is one folium. Okay. Now, if you take one folium. Now suppose you have taken this folium, okay? Now you have to identify the layers. So how to identify which is the innermost layer and which is the outermost layer? With the help of white matter. No, first of all you have to know where the white matter is situated, where the grey matter is situated, okay? So in case of folium, where the white matter is situated, the central core is made of white matter. तो ये देखो यहाँ पे ये white matter दिखाई दे रहा है. This is completely white matter. Okay, so that means this part will be the innermost part. So अगर ये innermost है, so this will be the outermost. This will also be the outermost. Okay. Now in the from outer to inner, three layers are there. The first is the molecular layer. See the appearance of the molecular layer. It is brightly stained. Okay. Why? Because we have seen that only two types of cells are there. Remaining completely it is filled by fibers. Okay. Next, here you can see the second layer, that is the Purkinje cell layer. ये देखो पार्किंजे सेल लेयर, तो पार्किंजे सेल लेयर में क्या दिखाई देता है, सेल बॉडीज ऑफ द पार्किंजे सेल्स. See, these are the cell bodies of the Purkinje cells. These are the cell bodies of the Purkinje cells. So, from cell bodies of the Purkinje cells, the dendrites they will go into the molecular layer, and axons will go deep into the white matter. Okay. So, ये हो गया Purkinje cell layer. Next, deep one is the granular layer. ये देखो, this is the granular layer. So, what will be the appearance of the granular layer? Darkly stained. ये देखो, ये वाला लेयर अपीयरेंस कैसे दिखाई दे रहा है, डार्कली स्टेन, राइट. कंपेयर विद दिस एंड दिस, ये डार्कली स्टेन है. व्हाई? बिकॉज क्लोजली अरेंज्ड ग्रेन्यूल सेल्स आर प्रेजेंट, एंड अलोंग विद दैट गोल्गी सेल्स आर आल्सो प्रेजेंट ओवर हियर. ओके, सो लाइक दिस यू कैन आइडेंटिफाई द लेयर्स. दिस इज आल्सो important. Okay, and I hope there will be no difficulty in identifying the slide. And in this particular folium, can you identify what is this? ये जो line दिखाई दे रहा है, yes, very good, it is pia metal. Okay, separate हो गया. Okay, next if you concentrate over here, this is the high magnification image. ठीक है, तो इसमें क्या दिखाई दे रहा है? First of all, here you can see these are the pyramidal cells. ये देखो, these are the pyramidal cells. ठीक है, pyramidal cells from here, if it is going towards the molecular layer, then these are the dendrites. ये देखो, these are the dendrites which is forming the dendritic tree in the molecular layer. Okay, next this is the granular layer where you can see densely packed cells can be seen. That's why it is dark in appearance. Okay, so same thing you have to see under microscope and then you have to identify. Okay, so this is very important. You can expect this one during the time of final practical exam. Okay. And lastly, there is a list. Yeah, here you can see this is the list of cells which are present in the cerebellum. Okay, here you can see this is the location of the cell, the feature of the cell, how does it appear. Okay, then the dendrites of the cells and the axons of the cells, where the dendrites of the cells are going and where the axons of the cells are going. Okay, so kindly include this chart also in your practical notebook whenever you are writing about the history of cerebellum.